Epigenetic reprogramming, especially through partial rejuvenation techniques using Yamanaka factors, represents a paradigm shift in how we think about aging, not just as a process of wear and tear, but as a programmable, reversible state embedded in the epigenome. This line of research is among the most exciting in modern biogerontology, as it targets one of the most upstream regulators of cellular identity and function, the chemical tags and structural modifications that control which genes are active or silent in every cell. Yamanaka factors, October 4th, SOX2, KLF4, and CMYC, are a set of transcription factors discovered by Nobel laureate Shinya Yamanaka, originally used to convert adult somatic cells into induced pluripotent stem cells, iPSCs. In this fully reprogrammed state, the cells not only regain the ability to become any cell type, but they also exhibit complete telomere restoration and a youthful epigenetic landscape. However, fully reprogramming cells also erases their identity, which is not desirable in vivo. What's particularly groundbreaking is that researchers have discovered ways to apply these factors intermittently or in limited pulses, what's now called partial reprogramming, to reverse cellular aging without erasing cell identity. In animal models, especially in mice, partial reprogramming has already demonstrated the ability to restore youthful gene expression patterns, improve tissue function, and, notably, extend telomeres. Studies published in recent years have shown that periodic activation of Yamanaka factors can rejuvenate organs like the liver, heart, and muscle, and in some models, has even extended lifespan. These experiments reveal that aging is not only a process of telomere attrition and mitochondrial decline, but also a consequence of accumulated epigenetic drift, a kind of molecular noise that causes the cell's gene expression to become increasingly dysregulated over time. Partial epigenetic reprogramming appears to reverse some of this drift. The reprogramming resets DNA methylation patterns, reestablishes histone modification profiles, and rejuvenates chromatin structure, all of which contribute to restoring a more youthful phenotype at the molecular level. Intriguingly, this also seems to restore telomere length, as telomerase becomes reactivated during the reprogramming process. This suggests that even in aged, non-dividing tissues, a window of rejuvenation may exist, one that doesn't rely on introducing external stem cells or gene edits, but leverages the cell's own latent regenerative capacities. One of the most exciting aspects of this approach is its non-destructive nature. Unlike full reprogramming, which effectively resets a cell to an embryonic state, partial reprogramming can enhance cell function and resilience while preserving identity and role within tissues. For instance, old muscle cells treated with partial Yamanaka factor exposure not only exhibited longer telomeres and younger gene expression profiles, but also regained regenerative capacity, healing more quickly after injury than untreated aged controls. Despite the enormous promise, the field remains experimental and significant challenges must be addressed. The biggest is safety. Improper or prolonged exposure to Yamanaka factors, particularly CMYC, can cause uncontrolled proliferation or tumor formation. Researchers are actively working on strategies to bypass oncogenic risks, for example, by replacing CMYC with less risky transcription factors or using chemical cocktails to mimic the effects without genetic alteration. Precise timing, dosage, and delivery methods are critical, and work is underway to develop tightly regulated systems that can turn reprogramming on and off with high specificity. Delivery remains another technical hurdle. Currently, most studies rely on viral vectors to introduce Yamanaka factors into tissues, which raises concerns for human application. However, next-generation delivery systems, such as lipid nanoparticles or mRNA-based therapies, are rapidly advancing, as seen in the COVID-19 vaccine platforms. These could allow safer, tissue-specific, and transient expression of reprogramming factors, greatly increasing the feasibility of eventual human trials. Importantly, epigenetic reprogramming is not limited to telomere restoration. It holds potential to improve mitochondrial function, reduce senescence markers, enhance proteostasis, and even restore youthful metabolic profiles. In essence, it doesn't just address one branch of aging biology. It may tap into the master regulatory system that governs aging itself. In the future, Partial epigenetic reprogramming could become a central pillar in anti-aging medicine, applied proactively to maintain tissue function and delay degeneration. One could imagine a future in which individuals receive periodic reprogramming treatments, either locally or systemically, to rejuvenate organs before disease sets in. As tools become more refined, safer, and targeted, this approach could offer a level of biological reset never before possible. In conclusion, epigenetic reprogramming through Yamanaka factors offers an astonishing window into cellular rejuvenation. With the capacity to restore youthful gene expression, extend telomeres, and revitalize aging tissues without loss of identity, it stands as one of the most compelling strategies under development today. While still in its early stages, the trajectory is clear. This is no longer science fiction. It's a rapidly evolving frontier, and it may fundamentally redefine our approach to aging, turning back the clock not metaphorically but literally at the molecular level.